Dr. Manary speaks from direct experience. He has dedicated his career to helping the poorest to overcome poverty and severe malnutrition. The Gates Foundation-funded Global Harvest Alliance that Dr. Manary helps to direct points out that, quote, over half of all childhood deaths are the result of an inadequate diet, with more than 3.5 million children around the world starving to death each year, and many who survive still face overwhelming challenges in the form of a lifelong loss of intellectual and physical potential. An article about Dr. Manary in the New York Times called Doctors Who Go the Extra Mile wrote, if you want to round in the pediatric nutrition ward at Queen Elizabeth Central Hospital in Biofortifying, the largest city in Malawi, you must get used to the constant wailing of children with kwashiorkor, a severe, often fatal form of malnutrition. For St. Louis pediatrician Mark Manary, the din is a cry for help he can't refuse. Since 1994, Manary has worked for months at a time with the malnourished children in this Southeast African country whose 10 million people are among the world's poorest. Tonight, Dr. Manary will speak on biofortifying crops to reduce food insecurity for the poorest Africans. Please help me welcome him to Oregon State.
Thank you, Steve, for that kind introduction. And I was looking over the website about all the different speakers you've had over the past few years, looking at nutrition, biotech, and all these issues. So I was actually quite daunted. I'm sure I don't have anything that that focused or expertise to say. But I do want to share with you how I got involved in BioCassava Plus tonight, and what BioCassava Plus is all about. And it's something that I dedicate a fair portion of my energy and effort to at present. But just by way of introduction to, I'm a pediatrician. I've been working in Africa the better part of the last 25 years. And I've been doing that, and as a person, I tend to be the person who doesn't approach a problem or a situation with a certain skill set or a certain toolbox and say, this is what I'm going to bring to the situation. But I tend to be somebody who says, whatever we need to learn, I can do that. I can still learn things. What is the elephant in the room? And certainly for kids in sub-Saharan Africa, it's being sick because you don't get enough to eat. As Steve mentioned, we're talking about over half of child deaths worldwide are simply because of that. Which when you hear all the stuff about AIDS, TB, and malaria, take those numbers, multiply them, add them together and multiply them by 3, and you'll get to about the amount that you could see the suffering caused by malnutrition. Another way to say that is listed out on this slide. We're talking about 83% of malnutrition-related deaths are not really in children who simply starve to death and then for want of the last drop of food die, but their bodies weakened, their immune systems weakened, they get common infections that become very serious, and that's what leads to their death and disability. We're talking about a world these days where 55 million children are wasted. Wasted means that you're acutely not getting enough to eat, such that your weight is relatively low compared to your height. When we talk about stunting, stunting means that you don't reach your physical or intellectual capability simply because you don't get enough to eat, or in more commonly because you don't get enough of the right micronutrients to eat. And we're talking about a world these days that happens about 20 to 25% of all kids. And of childhood disability adjusted life years, it accounts for 21%. Stunting alone does. So this is the elephant in the area of child health. And 17 years ago, when I went to Malawi, I said I was going to fix this problem for kids in sub-Saharan Africa. So let me tell you a little bit, just very briefly, about some of the things I've done, and then we'll spend quite a bit of time talking about what I hope to do in the next 10 years with cassava. When I went to Africa, I I'd been in Africa working for some years, but when I came to Malawi, it was clear that that was a place where the biggest problem kids had was they simply weren't getting enough to eat. And children were presenting to hospitals and being fed these milk-based formulas and being kept in these, crowded rooms for weeks on end. And really what was happening, which was following state-of-the-art WHO recommended standard care, was that about 25% of these kids were getting better. So after working in that situation for some years, I decided that we were going to try something way out of the box. We were going to try to treat kids at home, and we developed a peanut butter-based food called Ready-to-Use Therapeutic food. And this, when we first started this, it was was too far out of the box to be accepted, I think, by most people. Most people thought what we were doing was dangerous and cavalier and irresponsible. But as soon as we started doing it in 3 clinical trials that spanned over 3 years, what we saw was 90 and 95% recovery rates when we were seeing 25% below. So just sending children home with a formulated, ready-to-use therapeutic food and their mothers feeding them was better than all the care that the health systems and the doctors and folks like me were able to render to them. And you can see these series of recovery rates that we got there in Malawi. And this is just an anecdote of a kid who's very sick. This kid's got kwashiorkor. This kid is deathly ill from malnutrition. On the one over here, which I'm not going to hit the right thing, he's looking at about a 65% chance of dying. His mother feeds him this peanut butter food at home for 4 weeks, and he's fine. Okay, so this food is very, very powerful. Some of you may have heard of it. It's revolutionized the way malnutrition is taken care of. When we did these 3 years of clinical trials, 2001 to 2003, we realized this was on to something pretty important, and so we spent a lot of time, as Steve alludes to, with an NGO operationalizing this therapy and approach. And a good friend and colleague of mine spent a lot of time changing policy at the WHO in record time, and now this is what's recommended for malnourished children the world over. Okay, so that's rescue therapy. That's doing something to fix malnutrition for kids who already have it. But I really want to fix it. And I want to fix it by preventing it before it ever happens. Okay, so we're going to talk about some fairly less successful prevention strategies, and then we'll move into some agricultural interventions. So one of the first things people say when they say, well, these people eat all they eat is corn. They need to start being told to eat other things. They need to diversify their diet. And the idea that nutrition education is really going to be an effective sort of kind of intervention. Well, I have to say that my experience over the last 25 years with nutrition education is very disappointing. It's usually offered in very general terms because we want the intervention to be broad enough so that it can be, applicable to large numbers of people. But that doesn't give the individual a roadmap or a how-to of how to implement it. The best model of education is something called the positive deviance or the hearth model, which some people may have heard of, where in a given situation where you see well-nourished children and malnourished children in the same particular socioeconomic and environmental situation, you try to get the mothers from the children who are malnourished to learn something from the mothers who are well-nourished. And really the only success stories that positive deviance has to show are in societies where there are certain high nutrient foods, rich foods, that they don't feed to children, particularly maybe because they don't think children should eat them or they can't chew them or something like that. And the sort of, most successful story with that is feeding children shrimp in Vietnam. But in general, if you look at all the foods available, you can come to the conclusion that there's no particular advice you can give somebody for most of sub-Saharan Africa that's going to allow them to have a complete and adequate diet. And for those of you who who play around with Excel or linear programming or something, you can imagine how you could do this exercise. You can go to the market where all the foods are available, and really they're not too many, they're 50 or 60 only. And you have some kind of table with all the different nutrient contents of those foods, and you say, what would you kind of diet would you have to have in order to meet the requirements, the RDAs or the DRIs for these kids? And you say, oh, wow, so there's no combination of these foods that actually does that. So this is an interesting exercise, but I think what it's meant to communicate is that perhaps education has limits that it can't overcome. I mean, education can't, you can't tell people to do things that are impossible. So, and actually working in Africa, I have to say that this is not a surprise to me that education doesn't have many benefits in terms of nutrition, because the problem is really about poverty and it's not about ignorance. Oftentimes, mothers know that if they could feed their kid eggs. That would be great. It would be a great food. There's no way they can feed them eggs. So for people who want to fix this problem, looking into education, I became clear to me after doing some linear programming exercises the first few years that in Southern Africa, in Eastern Africa, there weren't options to create an adequate diet. So then we moved to supplements, okay? And probably the best known supplement program that around the world is a vitamin A supplementation program. So for 2 weeks every year, every UNICEF employee and lots of government employees in the developing world stop what they're doing and they go around and they stick vitamin A capsules in kids' mouths. And this massive effort, of course, is really somewhat expensive in terms of time and resources. It covers about 65% of the population, the target population that they're trying to cover. And although it's heavily backed by UNICEF and heavily supported by the vitamin A advocates, most people would say that this is not a sustainable solution that's going to be able to fix. And then if you say, well, what about iron, or what about zinc, or what about, I mean, you get yourself into a situation of very expensive supplements that just can't be rolled out on a broad scale. Food fortification, also another strategy to prevent malnutrition. So what we mean here, or most typically is in the US, would be exemplified by what we do with wheat. We put folate in our wheat flour. We can do that because most wheat is milled at some kind of central facility. So we don't have farmers growing their own wheat and then making their own flour and then consuming their own wheat. But most farmers in sub-Saharan Africa are doing that. So they're not sending out their flour for somewhere for some central processing. And when micronutrient supplements are simply provided to the home, the acceptance of that, surprisingly to me, is not as high as we might think. I mean, I'm thinking particularly about a case they had in Somalia. I mean, these were folks that were having their entire diet provided for them. They were refugees in a Kenyan refugee camp. They asked them to put a micronutrient sprinkles in their porridge. It was just there for the taking. There was nothing inconvenient about how this program was set up. And only 30% of the people would use that. And they said, well, we don't know what's in it, and we're concerned it might make us sterile, or it might be something these camp operators are trying to do to us. Which is really, really blew me away to think that even in such a controlled situation, we couldn't get very high levels of compliance with micronutrient fortification. And for those people who would say, well, they didn't do the right kind of education campaign and what, World Food Program learned from that and did a very extensive education campaign in subsequent camps and got uptake all the way up to 50%. So food fortification for sub-Saharan Africa doesn't appear to be on a large scale, a very viable program either, because all that would need to be put in to get public acceptance. So that leaves us, if we're going to passively prevent malnutrition, we're going to need to do something with agriculture. Something so that people just consume foods that provide all the nutrition they need, rather than having to be given a supplement like the vitamin A, or given something that they need to add to their food, or given advice that they can't follow, that kind of thing. And after developing these ready-to-use foods, I became convinced that agricultural kinds of interventions about 5 or 6 years ago were going to ultimately be part of the solution, and committed some of my time and resources to working on that. There's 4 sort of broad areas that quickly come to mind when people talk about agricultural interventions that are going to alleviate malnutrition. One of them is, very basic, and I don't think many people would dispute it: fertilizing and irrigating. Okay, these are things that are not done on a large scale in sub-Saharan Africa, and even when they are done, the results are pretty profound. You can double the output from a hectare with a cereal by simply fertilizing and watering without really any other specific kind of inputs. So these are things that need to be implemented. I think going along with what I said about nutrition education, one thing that's often advocated is dietary diversity. People should grow a more diversity of different crops. But they don't, this types of agricultural extension projects have been advocating this and promoting this and trying to do this with demonstration projects extensively for 30 years or so, and the populace isn't taking it up. And I think really why they don't is because there's something sort of fundamental in a simplistic way to say that if you've got a piece of land and you want to get the most calories out of it, the thing to do is to grow cereals, not to grow some cereals and some tomatoes. And so it's a hard sell, and it may not be the right sell to be promoting different diversifying of the crops. Commercial farming, of course, would be a lot more efficient, but in sub-Saharan Africa, we don't really have other ways to employ all the people that we're going to take off the farm. So if there was, then that would be a way of income generation and certainly economic promotion for those folks. And then biotechnology. So before I venture into biotechnology, I want to say a little bit about a traditional breeding project that I'll just ask, how many of you have heard of Harvest Plus? Some of you have. Okay, not a whole lot. So about 20 years ago, there was a very good idea by a group of agriculturalists that said, what we need to do is to categorize the nutrient content of all the germplasm of the staple foods of the world, and then maybe we can, crossbreed a high zinc line with a high vitamin A line, and we can improve the nutritional status of people all around the world consuming these staples. And so there was a really extensive effort about 10 years ago, around 10 years ago, to seek out for 13 staple crops the many thousands of samples and characterize their micronutrient content. And then the notion would be that once the right varieties, strains, species, whatever were found, they would then be bred to create very nutritious lines of staple crops. Unfortunately, in a very simplistic way, what they learned was there isn't that much diversity in the staple crop, in the germplasm and the micronutrient content. So there really wasn't any way to do that. It gets back to that same kind of ultimate wall we keep running into, that mass is conserved. If there's not those foods available in the market, you can't put them in their diet. If there's not that amount of diversity in micronutrient content in staple crops, you can't expect people to get adequate micronutrient intake from their diet. So this was a very disappointing thing, and now Harvest Plus has kind of coned in and refocused on a very few projects, and really, what they have to show to date is some orange flesh sweet potatoes going around Uganda and Kenya. So it doesn't appear that traditional breeding techniques are going to be the answer to malnutrition worldwide. And for those of you who are breeders, and I certainly am not, I'm told that it takes a long time to incorporate several traits. So if you need to have something that's, you know, dependent on 6 or 8 or 10 genes, that's going to be a many-decade exercise to create a line with all of those traits in them. And then, when consumers receive such a crop, sometimes they don't like them because they say, well, it's not what I'm used to. It doesn't look what I think cassava should look like. It's a hybrid. So it leaves us with biofortification. So what we mean, and I think everyone in the audience from what Steve, with all the different talks that have been given here, knows, it means this is using genetic engineering, modern science principles, to introduce traits, which can be the function of more than one gene, into staple crop lines. It's going to be a public health approach, you know, in other words, a population-based approach to micronutrient deficiency, rather than, you know, this sort of individual administration of supplements. Okay, so that would be sort of an active prevention strategy. This is the passive prevention strategy, which is inherently more effective. You can also incorporate traits that the farmer likes, like pest resistance or yield. And you need those kind of traits because inherently the farmer's not going to take them up so quickly if they don't have traits like that. Just to say that it's orange colored and that's going to be better for your kid's health isn't as compelling a reason as, oh my gosh, the brown streak virus doesn't kill it. And also then you can offer people foods and plants that look just like they've been eating for millennia. The other thing that biofortification offers is the opportunity to select targets for nutrient contents that will actually fix the problem. So if you're just breeding or using available foods, you're stuck with the levels of micronutrients that are in those foods. And, you know, if you don't, if that's not adequate, I mean, that's a limitation that you're still stuck with. Here, with the project we work with cassava, but can be for any nutrient biofortification project, we can set a target for a micronutrient content in that food that will meet 100% of their requirement or their RDA. And so, I mean, we've done that in the cassava by saying if a consumer consumes 25% of their energy as cassava, they'll get all the vitamin A, iron, and zinc, and vitamin A that they need. So right now, we have 14 million farmers in 25 countries that are growing genetically engineered crops. And 90% of those are smallholder farmers in developing countries. So as we know, there's some opposition to genetically modified foods. There tend to be development and political groups that oppose it, usually on the basis that the examples that we have of this are large agribusinesses that have then control of the seeds and the germplasm, which really kind of limits the applicability for the poorest farmers in the world. And then there can be concerns about gene flow. Maybe they would create a super species, or things will be forced out of the environment, loss of diversity. So I'll make a few comments about probably the best known nutrient biofortification project, golden rice, and then I'll tell you a lot about our cassava project. So golden rice, how many people have heard of golden rice? Everybody's heard of golden rice, great. Usually when people heard of golden rice, their first question is, why isn't it being used today? And so the reason to that question, the answer to that question is, it was on the cover of Science magazine 10 years ago when they had 6 times or so as much vitamin A in the rice, and it looked different color, and it made a nice picture, and it demonstrated something important about biotechnology, but 6 times the increase in vitamin A would do nothing for human health. I mean, you need 60 times in order to get any kind of health impact. So golden rice has been around, and people have learned about it and known about it for a while, but it really wasn't until about 2005, 2006, after Syngenta got involved, that they were able to get, such high levels of vitamin A in the rice. So I think I just said most of what was on that slide, but they started out with some daffodil and bacterial genes, and then when they replaced them with the maize gene, that's when we got much higher levels of expression. But golden rice right now is in the field in the Philippines, and in 2011 it's going to be out with the consumers. The studies that have been done with consumers in the Philippines seem pretty positive. The consumers are looking for it. They're interested in it. The small acceptability trials have gone very, very well. The amounts of vitamin A in golden rice should make a very significant impact on vitamin A deficiency. And I didn't sort of belabor the point, but vitamin A deficiency is one of the micronutrient deficiencies that's sort of the very best categorized or cataloged in the whole world, accounting for hundreds of millions of adjusted life years annually. Of course, in a very extreme case, it causes blindness, but that's not really how it usually presents. It just usually presents as an increased morbidity or mortality for children under the age of 5. So, biotechnology with golden rice. And thus also done human trials. I think people might have known that. They did a human trial in China, and they did some in the US with golden rice to demonstrate actual efficacy. So it's not simply an idea. We have this plant. The plant demonstrates this trait. It'll do this. But they've actually done trials with humans that show that they do get better vitamin A status when they consume golden rice. So the project I work with is called BioCassava Plus. And this was born around the time of the millennium. And at that time, the Gates Foundation was seeing themselves as one of the major organizations that was going to impact human health worldwide. And they issued 14 grand challenges. And it took them a few years to formulate these challenges and then accept proposals and then choose proposals to fund. But one of the challenges was create the perfect staple plant food. And those of us who work with cassava formed a big tent team, which basically includes most anybody in the world who works with cassava, to make cassava a nearly perfect staple food. And after 2 years of, making it to the next cut of proposals and rewriting something or another, the Gates Foundation in 2005 announced that that was one of their chosen projects. And for those of you who do science research, this was a very interesting introduction that they gave to us. Usually when you want to do a project, you outline what you're going to do, you have a methodology and a protocol, and you get a certain amount of funding, and that's what you do. And the results are what the results are. The Gates Foundation said, no, in this particular project, money's not an object and failure's not an option. You need to figure out how to make this work. So rather than sort of laying out a strategy that you think might be the most likely way to increase vitamin A in cassava, you need to lay out 3 or 4 different pathways, start on all of them, and when one looks like it's going to be successful, stop working the other ones. So it's not a typical kind of science-based research project. We chose cassava because it's the food of the poorest people in sub-Saharan Africa, so it's what they eat right now. Provides about 35% of all the calories consumed in sub-Saharan Africa. And here we show people processing cassava. Processing cassava because it has toxic cyanide in it, and they need to do these things to get rid of this cyanide. We'll talk a little bit more about that. We chose cassava because it's not very fussy for its soil or water requirements. If the weather turns bad, the cassava stays in the ground and you can harvest, it just stops growing, but it doesn't die, and it'll start growing in a couple months when more nutrients are available. It's got this peel. It's like a potato looking thing. And this peel is just chock full of cyanide, I mentioned. And so nothing wants to eat it. Worms, rodents, they just leave cassava alone because it kills them. There's not much weeding that's required, and it can be planted at any time of the year, and it can be harvested at any time of the year. So it usually matures, say 9 months, 15 months after you plant it. And you can just take it out of the ground, and you do, usually within a few days of when you want to eat it. Now, the limitations of cassava are that it's the lowest protein energy ratio of any staple food, period. 1%. For those of you who are familiar with those kind of figures, you'll know like cereals are sort of 7 to 15%. So cassava is a very low protein food. It's, as I said, these cyanides in the peel are great for protecting the plant, but if you don't leach them out in the food processing during food processing, you can be left with a permanent paralysis below the waist. And I remember when I first went to Tanzania, I hadn't been there very long. There was a village that was nearby. There was a wedding. They needed to prepare a lot of food in a hurry. Cassava was their staple. And we were called to see 128 people who were left permanently paralyzed below the waist because they hadn't taken the 3 days process. So the cyanide, even though it isn't such a public health problem, can be devastating when it hits. And cassava, needless to say, I said it doesn't have any protein. It also doesn't have any micronutrients whatsoever. It doesn't have any vitamin A, has very low levels of zinc and iron, which are the micronutrients we most often talk about. So when we put our proposal in to the Gates Foundation, we said we are going to provide enough zinc, iron, protein, vitamin A, and vitamin E in cassava so that if you're a staple consumer of cassava, you'll get 100% of that. We are going to do something to make it less toxic to the consumer so they don't have to process it quite so much. Now, one of the features of cassava is that when we talked about just taking it out of the ground whenever you want to eat it, well, people do that because it rots really, really quick. So in 2 or 3 days, a cassava will be like a sponge. Usually it's hard like a potato when you first take it out. And it would help the farmers if it didn't rot quite so quickly. If they had a week or two, they might be able to move that cassava to a market and sell it rather than just consume it as a subsistence guard. And then, like all plants, cassava is susceptible to viral diseases. And I'm not sure people are familiar, but there's a very bad virus infection going around East Africa right now, brown streak, which has taken yields of smallholder farmers down by a factor of 50. Okay, so essentially made so they're not growing any cassava. A drop in 98% of their yields in infested fields around Lake Victoria. And then, of course, biotechnology with plants isn't all about hand waving. I mean, for those of you who are familiar with how many drugs are made today, bacteria that are engineered to do this. And the bacteria, for instance, like insulin, I mean, the bacteria has no interest ever in making insulin, no use for insulin, but you put the insulin gene in there and you make a lot of insulin, and it's made a lot cleaner and a lot safer and a lot less messy than the way we used to get insulin was, taking carcasses from pigs and cows at slaughterhouses and extracting the insulin. So, but when working with the plants, one thing I've learned is that it's not like bacteria. You most of the time, if you decide to put a gene in there, all you get is a plant that doesn't survive, kind of thing. Really, we've found in the BioCassava project the most successful avenues are when we copy successful strategies that we see already in nature. So let me tell you a little bit about the progress to date with BioCassava Plus. This was about 5 years ago. And it was thought to be a very, very ambitious project. And I remember people saying that was, with a smirk, that was going to be too ambitious. We're not really going to be able to do that. And I also remember people saying that when we did home-based therapy, so I didn't really take it all so seriously. But our goals were to increase iron and zinc sixfold. And the way we attack the, first of all, iron and zinc deficiency worldwide are huge public health problems. I think iron deficiency affects about half of the world's population. And if most of those people aren't in Europe or North America, then you can imagine it affects a majority of people in sub-Saharan Africa. Zinc deficiency is the second largest nutritional problem in the world. Our strategy for iron was to, what we need to do is we need to not just get more of these kind of nutrients just generically anywhere in this plant, but we need them stored in the root and kept there, so when the consumer goes to eat, they have availability to that. So we need to use tissue-specific promoters. We need we're using potato root promoters that will express these genes only in the storage root, in the tuberous root. In iron, we're using a very avid iron transporter from Chlamydomonas, an algae that lives in the middle of the ocean. And, in the middle of the ocean, you can imagine you have to have something that's very, very avid to suck up a divalent cation, and very, very selective, so it won't take up another one like, say, cadmium or something like that, because we don't want to create plants that in some way have toxic amounts of some kinds of nutrients. And so this iron transporter FEA from Chlamydomonas is used to bring the iron into the root, and then we need a sink, someplace to store the iron, and we're just using ferritin, which is a protein that's found in many plants and many animals. In zinc, we use a strategy that we use a series of 2 transporters, one to bring it into the root, and then one to pump it into a storage vacuole. And as far as what we've learned so far in the first 5 years with this, the iron transporter works well, and we can get 6-fold increase in iron in cassava growing in Nigeria and Puerto Rico. We can't really achieve such high levels with zinc, and zinc is the one trait that we're not going to get on our making a perfect staple plant food. This graph here is meant to show the same kind of thing. That's wild type, and you can see the iron is 2.5 times higher, and that iron is about 3 or 4 times higher, and that it's concentrated only in the roots, not in the other parts of the plant. As far as protein goes, the strategy that we found that was successful for protein, and protein was a harder biological problem to attack, because the plant only wants a certain amount of protein in its cytosol. And so just sort of putting genes in and telling it to make more protein doesn't do anything other than make the plant unable to survive. But we noticed that in sorghum that these inert protein granules can be created with a 33 amino acid leader sequence, a Zane sequence it's called. And it just stacks these proteins in these very, very tightly together, and then they're sort of metabolically inert. And so if we put that leader sequence on a protein that we want to overexpress, and we've been using a sweet potato protein, sporamine, we are actually able to get five-fold protein concentration in the cassava. And this visually shows you that. This dark staining is for protein, so the wild type doesn't have very much. And then you can get, with the patatin, the root-specific promoter, quite a bit. And it works with any kind of protein you want to try, at least the 6 or 7 that we've tried. So you can choose a protein that has a good amino acid complement, and it's going to be best for nutrition. In terms of vitamin A, our goal was to get a 50-fold increase in vitamin A concentration, and then not to decrease the vitamin E and I can tell you a little bit about the pathway on that. So typically, this is something you don't need to know, but it's a biosynthetic pathway for vitamin A and vitamin E, and they're related. And so if you're often driving, like golden rice is driving phytoin synthetase, well, one of the problems they had was very low vitamin E levels, so they're driving, the product out through the carotenoids, and the tocopherols are not in the same, are being in reduced quantities. So to overcome that, we decided to overexpress 2 genes in the pathway up here. One of them is just going to increase the precursors for both vitamin A and vitamin E, and then phytoin synthetase. This is a same maize gene that they're using in golden rice. And we can get, I think 60-fold was our best line for vitamin A so far, growing in Puerto Rico, and it's also growing in Nigeria. If we don't use both genes, we get less vitamin A. This slide here is just meant to show you that the double construct vitamin A gives you up to 60-fold more of the vitamin A in cassava. And the vitamin E does not go down. There's your wild type and 3 lines that we have. We started looking into the post-harvest deterioration, the rapid rotting of cassava, by trying to look at what the oxidative signaling pathways were. And that didn't seem to yield anything very interesting, except if you messed around with that, then the plant wouldn't grow at all, would never form roots. It didn't do anything. We had a colleague up at Washington State working on that. But when we have higher concentrations of vitamin A, we do get extended shelf life, if you will, or less rapid deterioration of the cassava. So it's kind of a dual benefit of increased carotenoid content. You can see here, this is 60% of these things are after 7 days of storage, were rotting on the wild type. But down here, in these lines, when we had carotenoid concentrations, almost what we would consider our target. Our target's 40. We have almost no rotting after 7 days. Some pictures of orange cassava. Okay, so at BioCassava Plus, we're excited because we've hit most of our, I didn't show you any slides about the virus resistance, but we have some success with protection against Gemini virus and brown streak virus as well. So for our goals for making cassava a nearly perfect stable plant food, we're able to nutrient enhance it with protein, vitamin A, maintain vitamin E levels, iron. We're not really able to do zinc, and we're able to delay the post-harvest deterioration, and we're able to induce some virus resistance. So we're feeling very successful, and so is the Gates Foundation. We've moved ahead to a second phase of the project. We chose initially target countries Nigeria and Kenya because those are going to be the places where we're going to have the most impact. You know, everything needs to be deregulated and introduced to a national agricultural service, and so Nigeria is just a big place. There's just lots of cassava consumers over there. So that's one of the reasons why we chose that. And of course, this lab work that I showed you is lab work, greenhouse work, some stuff we sent to the field in Puerto Rico. In order for it to be useful to farmers, we need to be able to transform local varieties that they want to consume right now, that they are growing right now. And in fact, we have been able to. We've been doing that for about a year, picking out the most commonly used varieties in Kenya and Nigeria, and we've been successful. Nigel Taylor's done that over at the Danforth Center in St. Louis. So we feel like all the pieces are in place to move ahead and try to do some regulatory field trials and grow these plants in Africa and see how well they fare there. When we do that, we have to do a careful assessment, environmental risk assessment. Okay, and I have a few slides about sort of the general principles that are used for that, and we're in the process of pursuing that there. We have permits, we have plants, we have things growing, but we don't have data yet from Nigeria or Kenya. And when doing this, people best qualified to come to accurate conclusions are people that understand the biology of the crop, the introduced trait, the environment that the plant is growing in. And we're always comparing it to what the wild type cassava does. So, you know, in order to bring this crop, to let this crop out of an experimental field, it's going to need to perform just like the cassavas that are growing there right now. So it can't be shorter or have a different dry matter weight, or can't have a different branching pattern. One of the things that really helps us, another reason why we chose cassava, is in terms of gene flow. You know, cassava is vegetatively propagated. So the way you get more cassava plants is you cut off the stems of cassava plants you have and you plant those in the ground. And so it doesn't make seeds. And the gene flow element is really, really, really limited, which is different than, say, something like a cereal like a sorghum or something, which will have some gene flow to wild relatives. In terms of environmental risk, we want a thorough characterization of our GM plant. So we don't want just to say that it has more vitamin A, but we need to know about all the different hormones in that pathway and the levels that they're at. We, as I said, are monitoring these plants in 20 or 30 different parameters so that they need to perform just like the wild type plants, looking at the phenotype. It's not really so important about where the gene is in the plant and that kind of thing. It's more about what kind of phenotype they're going to express. And this cartoon, I think, looks at the different elements that we're looking at. And compared to other kinds of GM plants, ones that have insect resistance, you need to be able to look and see about where there's increased potential for the insect population or non-target organisms and virus. Will there be viral resistance and weediness and invasiveness, altered fitness, herbicide tolerance? But down here with nutritional enhancement, we don't have models from industry or from other folks as to what exactly to look for. So we're looking for that there's no differences, of course, in their insect and virus resistance and altered fitness and herbicide, but it doesn't feel like there would be a selective advantage in any way for a plant to have enhanced nutrient. I mean, you can imagine that in some way that nutrient might be more attractive to something like birds that want to eat this plant, but nothing wants to eat cassava. It's got all that cyanide in the peel. So we don't really see that there's any particular dangers along those lines, or the cassava is not really able to transmit those genes to other cassavas that it would be growing next to. And then to keep belaboring this point a little bit is we're looking for no unintended changes, no intended toxicity to a target organism, and no increased fitness, no changes in agronomic impact and practices. This comes over years of field trial experience. So we don't get this by planting one crop in Nigeria and assessing it. I mean, we realize that before people in Africa, if they're going to realize some benefit from BioCassava Plus, that it's going to be at least another 5 years and 6 cycles of planting to do this. But we want we don't want to introduce anything that's going to be bad for people or bad for the environment. We're trying to fix malnutrition. So that's a little bit about the cassava. And of course, I think we're going to have a time to answer questions. Be happy to answer more about it. Just as we finish up the talk here, I mean, I think that a few general comments about biofortification, and then a sort of a very general comment about feeding the world. We see most of the costs with biofortification coming on the front end with research. And we also see, though, that the value within this BioCassava Plus 2, I mean, the people scientists, junior scientists doing this work are from Nigeria and Kenya, setting up labs in Nigeria and Kenya where at their national agricultural institutes, they can continue to transform cassava. So that is very important to us, because because we've chosen a couple of varieties from each country and we can successfully do something in a lab here in St. Louis isn't going to change it. We need to transfer that technology so the folks in Nigeria can use it whenever they see and that it's important. These are public projects, so nobody owns them. Nobody owns these plants. These plants can be used by anybody. These plants can be manipulated by anybody. The beneficiaries, of course, are people who don't buy these plants, products. They're subsistence farmers, and they're going to be able to vegetatively propagate them for some generations. The reason I'm involved with biotechnology is, you probably could get the flavor the way I led up and talked about other kinds of interventions like supplementation and fortification and so forth, is because I don't see, without major advances in agriculture, how we're going to feed everybody when our population grows 7 and then 9 billion, which is what we're looking at here in the next generation or so. We can't create more farms. There's not a lot more arable land to open up into farming. Right now, we're kind of at a break point in terms of how much food is produced in the world and what the nutrient needs of the world are. We don't have an economic or a transportation system to deliver it to everybody in 2010. But as the population grows in this world, and it is, even though it may not be increasing at what it has been, agriculture needs to make some quantum leaps. So I don't know everything, or very much actually, about how to fix this problem of feeding the world. But I do know that there have been a couple instances in history where there's been some pretty unexpected turns of events. For the sort of nihilists and those folks that would say, well, we're just doomed, people are going to starve, it's going to be ecological disaster, and so forth. I think about, those hunter-gatherers in Africa in 1600 who were pushing the carrying capacity of their land. Some slave traders from the New World came over and kidnapped enslaved some people on the coast, but they also introduced cassava and corn, and these crops spread across Africa and converted hunter-gatherer societies to agrarian societies, and agrarian societies can feed 10 times as many more people on a piece of land as a hunter-gatherer society. And so when Africa didn't look like it could feed itself in 1600, it was then able to with the introduction of agriculture. And then I think for the older folks in the audience, we remember in the 1960s and 50s thinking that India could only just be a place where everybody starved every other year, and what could ever happen in India would be good. And then we had this Green Revolution, which I'm sure most people know a lot about. But one of the essence of the Green Revolution, of course, is selecting plants with larger edible parts, and that, with irrigation and fertilizer, converted India to a food-sufficient place. So I don't think it's impossible that we're going to feed a planet with 9 billion people. I do think we're going to need technological advances in agriculture, and I do think biotechnology holds some promise in that. And more than just making general statements, I see that BioCassava Plus could make a huge impact in Africa. So it's about what I have to say, but I think we'll have a lot of time for questions now.
Have you done any animal studies with your biofortified cassava now in terms of nutrients?
He asks, I'm supposed to repeat your question. He asks that, have we done animal studies with the biofortified cassava? So, no, we haven't, because we've done human studies. So we've fed healthy volunteers in St. Louis. There's so many limitations with animal studies, and and so much uncertainty in it, we thought it wasn't really worth the trouble.
And what are the results of
the iron, vitamin A, and protein is digestible, bioavailable. You can measure that that this is getting into the folks, just like, say, a vitamin A rich vegetable like sweet potato or something. So they've been good, yeah. Yes, ma'am.
I have a question about the cyanide content
Yeah.
in the BioPlus. Is it reduced?
Yeah, I didn't mention that. So when we looked into that, our first idea was that maybe we could help ourselves with protein by diverting some of the nitrogen that's going into cyanide into amino acids. But when we did something to manipulate the enzymes in that pathway, we were unable to. The plants wouldn't form any roots at all. So the plants seem to want levels of cyanide like they're making in order to develop normally. So what we have put in is a lyase, which is an enzyme which will facilitate the breakdown of cyanide. So when those women are needing to do that for 2 or 3 days, beating around on it and drying it, now they only need to do it for 2 or 3 hours. But they still need to do it.
I have one related question. So is it cassava is a crop which is grown alone, or is it a practice of mixed cropping that other plants are simultaneously being grown along with the cassava? And if cassava can protect itself from attack of pests and other things, it may be also beneficial for the plants which are growing along with it. So in that respect, maybe having cyanide in the cassava may be useful.
Yeah. So she asks, is cassava grown alone, or is it intercropped with other things? And could the cyanide that's in cassava have a protective effect on other plants it's grown with? I would say in practice, in places where I've been in Africa, it's usually, there's usually patchwork, so there's some cassava and then some cereal, and but not sort of intercropped like we see corn and peanuts all the time. It's sort of a long straight stalk, and then it branches into a lot of leaves about so high, taller than a person, 6 or 8 feet.
I'm wondering about the accumulation of nutrients in the cassava. Like, does that deplete the soil over time, or how does that work?
No, in general, these soils have got ample amounts of iron and stuff in them. So we're not going to deplete the soil of iron by making this go from 5 parts per billion to 40 parts per billion or something like that, which is the kinds of increases we're talking about. Carotenoids are synthesized, so that we're not depleting anything of that. And zinc also is pretty prevalent in the soil. We haven't, it's an interesting question, which I don't really have anything useful to say about, is that, how far could you get by ignoring the plants and just trying to improve the soil? But it's an interesting question. May have some benefits for creating more food. Yes, ma'am.
I'm interested in, you said that it was a staple crop, and so therefore it was in public domain. Yet Monsanto has taken the staples that we thought were in public domain and patented them. Now, is there any fear that Monsanto or someone like Monsanto will come along and patent your cassava?
No, it's not that. You're right that she asks if they could a patent. Could patent be used to remove bioengineered cassava from the market or subsistence farmer market? The fact that it's a staple doesn't protect it in some way. The fact that it's a staple, what I meant by that is that it can have a broad impact on the population. And we have secured agreements or one of the fundamental elements of working with the Gates Foundation, for any of you who have ever done that, is that there's no intellectual property. It's all in the public domain, and it's all written out, and all those, they have lawyers that triple check all that kind of stuff. So there's somebody on our team who just does IP and works closely with the Gates Foundation, and they tell me that's not possible with the stuff we're doing there with the cassava. So I trust them, I guess. you know, in general, I don't really know the mind of Monsanto. I mean, they're not people I've ever actually been into their company or anything, but I mean, they're interested in they're a for-profit company, and so subsistent farmers in Africa, I mean, are an attractive market to them. Yes, sir.
Once you've gotten it approved, how will you mass propagate it to the farmers?
So we asked once if it were approved and shown to be effective, how would we mass propagate it? So we're going to need some kind of facilities, I guess if you want to call it that, where we are growing the cassava, these improved varieties of the cassava, and then distributing them freely. You know, so these would be like nurseries, you could think of them that way, or these, I don't know if any of you have been to East Africa and seen these banana propagation facilities, but we're going to have to do something like that because we, you know, they're not seeds we can distribute, so that we can distribute just, you know, shoots to the farmer.
Given the reservation by lots of people about genetically modified foods, A, do you have any evidence, or any evidence at all that this is bad or that there's some implications for human consumption? And B, what do you say to those folks?
So at this point, I have to, you know, say that our information is incomplete. That's why we need another 5 years of field trials. But at this point, we don't have any indication that these plants are abnormal anyway, or have any, the gene flow seems to be occurring, okay? For folks that say that, I say, that's great, don't eat them. But there's really no reason that you should, unless there's a danger to society, a genuine, you know, measurable danger to society as a whole, there's no reason to deny people the right to getting adequate nutrition on the basis of, you know, sort of something that's more faith than science.
This is an interesting crop because of its crop. It doesn't, you know, have seeds and mix with the other varieties. Have you worked with other plant materials that have those kind of properties where airborne seeds or pollen or something?
You know, I haven't personally, but people that I work with have worked with sorghum and rice and banana. Banana's another sterile dead end product, so that's a good one for this kind of target too. So there are different challenges. That's why I say in this environmental assessment, it has to be on a case-by-case basis. It has to be on a crop-by-crop basis. You need to look broadly. You need to look at you know, what's happening to the insects and the birds. You know, maybe we're lucky and cassava's going to work, and because it has all these benefits. Yes, ma'am.
I'm a vitamin E expert, and the tocotrienol is not actually a form of vitamin E that the human body makes, or uses rather. And what I'm wondering, how difficult would it be to saturate the tail to make an outfit tocopherol, which is the form that humans use?
I don't know. You know, our interest in vitamin E is more from the point of view of how it preserves the plant, and seeing, you know, deleterious phenotypes in the plants with lower vitamin E's. So, thanks. That's a good idea. I haven't been repeating these questions though. Steve.
I've got a question for you. So the environmental assessments you were talking about, where pretty much to get through the filters you expect to face, I guess both in field trials and ultimately in commercial use, things can't change apart from the target traits. That you had slides talking about this needs to be the same cassava. And it just seems that given how ambitious this project is, you know, you're really trying to change a lot of the physiology of this plant, that things won't be the same. And it seems that, so my questions are twofold. Given that I'm right, and that you will have a green revolution crop that's semi-dwarf, is physiologically different in many ways. It's not just that it's short. how will it get through the regulatory pathway? And then in the countries where you're working, do you know what the pathway is, or is it really still undefined?
Okay, so the question is, if you try to change lots of traits, you're bound to change the phenotype of the plant. And then how do you take a plant with a different phenotype through a regulatory approval process? Okay, so we've been operating under the assumption that we need our plants to not have phenotypes that vary outside the norms. Now, you know, this is a statistical issue. So there's a variation of, you know, the plant heights, or the height when the first branch comes off, the color of the petula, and these kind of things. We haven't seen, you know, that we've put protein and vitamin A together in a dual construct. We've put iron and vitamin A together in a dual construct, and we haven't seen phenotypes that vary outside of that range. So they're not the same dead on, you know what I mean, like the same average height, but they're within that range of what is normal. We do have we realize this is a changing environment, you know, a changing landscape, excuse me, for regulatory. So we know that the rules can be changed on us at any given time. But we spend quite a bit of effort learning what the regulatory landscape is, and particularly, I mean, we were the first GM crop approved to be grown in Nigeria, was BioCassava Plus. We understand what they're looking for. You know, actually, we've been really, really, really pleasantly pleased or surprised by the response of the national regulatory boards in Kenya, Uganda, and Nigeria. When you engage the health people and the economic people, they provide a strong argument that they're really interested in this stuff. And so they kind of bringing to the table, not just narrowly hanging in there on the agriculture folks. Yes, ma'am.
I'm curious, you were saying working with the Gates Foundation that they wanted you to take an approach where you would have multiple ideas that you're going after at the same time. How much do you feel this has sped up the process?
Yeah, so the question is that since the Gates Foundation encouraged you to take multiple approaches to answer the same question, how much does this speed up the process? I think the answer's got to be tons, because, you know, for those of you who have good ideas, I mean, I would say that when I have a really good idea, if I have 10 of those, maybe one of them works. So you can imagine if we've got this big tent cassava team, which has got, you know, people from all over the world, from China and different places in North America and South America, you know, we're dealing with people with a lot of experience and a lot of pretty strict, you know, the pretty firm ideas about things that don't agree. And so having all these different, allowing us to pursue different tracks at once meant for more harmony in the research project. I mean, you're going to be judged by basically the results you get. You know, does your cassava work kind of thing, rather than whose idea is better than the others. And I think it allows for, I mean, they're really in a hurry, the Gates Foundation. You know, if you tell them you can do something quicker if you do it, you know, if you have $1 million more for a year, they love that idea. they just really...
What is the process of harvesting the cassava to getting it to the family dinner table?
The question is, what's the process of harvesting cassava to the family, taking it to the family dinner table? So, first of all, cassava is, because it rots so quickly, is usually consumed either by the person, you know, by the family that harvests it, or their neighbor or something, you know, very, very, very close proximity. You take it out of the ground and you wash it, you break it open and you know, break it up a little bit mechanically, dry it in the air, which lets the cyanide evaporate and break down. And then Mostly what people are doing, one of two things with it. They're forming a flour and then cooking something, you know, like a wad of dough with it, or they are just boiling the cassava outright, you know, so just peeling it and just boiling it.
So is that a process of spices then? I mean, the flavor, what's the...
The flavor. Food in Africa is really bland. It really is bland. Yes, yeah.
Is it the same as yuca? Seen in Latin America?
I don't think so, no. It's not. Cassava is not the same as yuca. Yes, sir.
So is there a combination of fertilizer and GM plant that maybe you could bring to fields more quickly and would be more effective, or have you focused all of your research on just developing a plant?
Yeah, the question is, have we focused on all of our research on developing the plant, or have we looked at the role of fertilizer? And we have focused almost entirely on the plant, you know. So we want a hardy plant that we can throw out there in the worst environment, and it's still going to win.
Thank you very much, Mark.
Thanks, Steve.